Hi, my name is Anye. Here's my ID. So basically today I'm going to explain two different mechanisms to where T cells can become activated. So the interesting thing about T cells is that they're pretty picky and they want their antigen to be presented to them in order for them to elicit any type of response. So basically what happens is that you have a couple different situations. You have some sort of a macrophage or some sort of a cell that has been invaded by bacteria or infected by bacteria that has bacteria that has infected their DNA and their nucleus. So basically what's going to happen is within the nucleus, you're going to code for bacterial-like proteins. And so what happens is that the proteasome of the cell is going to recognize that being red flagging those proteins that, this is not my regular proteins. These are proteins that will come from an infected cell. So the proteasome goes ahead and breaks down those intracellular antigens, antigen proteins, and basically imports them into the ER. The interesting thing about this is that we also have the MHC-I molecule. So the MHC class 1 molecule is the major histocompatibility complex, and that's what antigen presenting cells like macrophages use basically to put on their surface and invite or recruit T cells to recognize those signals or those proteins. So basically what happens is that, like I said, the proteasome goes ahead and breaks the intracellular antigen that's been coded by this virus or bacteria that has invaded this cell. And it puts it combines a fragment, nonopeptide of antigen protein onto the complex and goes ahead and places this, if you can see here, it goes ahead and moves this complex into the Golgi. So the Golgi packages it into a vesicle, and it basically puts this complex outside of the macrophage, or APC for short, antigen presenting cell, so that they can present this to a T cell. So the type of T cell that recognizes this MH1 class 1 cell is a cytotoxic T cell. So basically it's telling the T cell that, hey, I'm ready to die, I'm invaded, I'm compromised, so go ahead and kill me. And the cytotoxic T cell, or the CD8 cell, will go ahead and destroy this cell. So the other class of major histocompatibility complex is class 2. And basically what happens is that you have extracellular antigens that the macrophages go and eat, because macrophages just love to eat anything. So they go ahead and eat that thing. They recognize it as foreign. They'll go ahead and break it up, put it into something called an endosome. The endosome moves into a lysosome, forms a phagolysosome. Okay, so the phagolysosome breaks down this antigen into little pieces or peptide pieces. And the similarity between the MHC class 1 and MHC class 2 is that you have the major histocompatibility complexes are originated inside of the endoplasmic reticulum. So that's the site where the broken down protein of this antigen will combine with the MHC complex. And so again, another similarity is that the Golgi apparatus is where the major histocompatibility complex is wrapped around by this complex, and basically is packaged to be set in a vesicle and to be put outside of the cell. So with this cell, with this MHC class 2 APC cell, basically it recruits CD4.